Hi, I'm Siti. Hi, I'm Anya.
Hi, I'm Faris.
Hi, my name is Mastu.
Hi, I'm Lina.
And this is our assignment videos on application of NGS technology in forensic.
In forensic investigation, DNA analysis provides irrefutable evidence that transcends the limitation of traditional investigative methodologies. It has revolutionized forensic science by offering precision and reliability in identifying individuals and linking them to crime scenes. Moreover, DNA analysis is important in resolving cold cases and exonerating the innocent. Investigators can link perpetrators to multiple crimes or absolve individuals who were wrongly accused by comparing DNA profiles stored in forensic databases. Therefore, DNA analysis in forensic is undeniably important, serving as a definitive and scientific method for establishing connection between individuals and crime scenes, while also ensuring the integrity of the criminal justice system.
So, what is NGS? NGS stands for Next Generation Sequencing that evolved from Sanger's chain termination in 1977. NGS method allowed the rapid and parallel sequences of millions of DNA or RNA to know the information on ancestry, paternity, and phenotype. In decades, we can see that NGS has become essential in various areas like biotechnology, virology, and medical diagnostics. Fun facts: traditional sequencing methods mostly rely on sequential processing, making them more labor and time-consuming. NGS has revolutionized genomics by significantly increasing the speed and efficiency of DNA sequencing, making it essential in various fields. One advantage of NGS is speed. NGS is relatively faster than traditional sequencing method. NGS technologies offer sequencing of billions of DNA in parallel, enabling analysis of multiple samples simultaneously and minimizing the need for fragment cloning method often used in Sanger sequencing. This is crucial in forensic investigation where timely results are essential. Second, NGS technologies is cost effective. Although the initial setup cost for NGS can be relatively high, but the cost per base has significantly decreased over time. This is due to the capabilities of NGS technologies that allow forensic laboratories to maximize resource usage by processing numerous samples in a single run. The reduced time, manpower, and reagent in NGS means that the costs are much lower. The third advantage of NGS technologies is that it is accurate. Repeats are intrinsic to NGS, as each read are amplified before sequencing. NGS allow for more repeats than Sanger sequencing due to its speed and cost effectiveness. The increased repetition enhances coverage, resulting in a more accurate and reliable sequence.
So in this part of the presentation, we are going to talk and explore more on how actually does NGS work. How do we prepare the sample, how do we analyze the data, and how do we sequence it. So in principle, the concept behind NGS technology is actually similar to those in CE sequencing. But the only main difference is that in forensic genomic applications, we only use forensically relevant sequences through an amplicon-based workflow. So in NGS workflow, there are 4 basic steps. So the first step in NGS workflow is library preparation. So we start with 2-step application with the first PCR step using sequence-specific tag primer pairs for each forensically relevant sequences in the DNA sample. So following this first step is incorporating the indexes and adapters into the DNA samples. Then we are going to purify the library and pull it into a single tub and linearize. So the next step in NGS workflow is the cluster generation. So from the library that we have prepared earlier, we are going to inject it into a flow cell where fragments are captured on the surface-bound oligos complementary to those on the library adapters. So now each fragment are then amplified through bridge amplification and the targeted sequence are ready. So now So the next step in NGS workflow is obviously the sequencing. So we use reversible terminator-based method to detect single bases as they are incorporated into the DNA template strands. Last but not least, the last step of NGS workflow and the grand finale is the data analysis. The sequence reads are aligned to a reference genome. After alignment, we can proceed with multiple analysis such as STR typing, mtDNA analysis, phylogenetic analysis, and so many more. So these are all 4 basic steps of NGS workflow. There are library preparation, cluster generation, sequencing, and data analysis.
The first application of next generation sequencing NGS in forensic is short tandem repeats, or also known as STR. STRs are specific sequence of DNA that are short, typically consisting 2 to 6 base pair that are repeated in tandem along DNA strand. Using DNA to distinguish between 2 individuals is a tricky method. This is because 99.9% of our DNA is same as everybody else's DNA. The analysis involves determining the number of repeats at a specific STR loci of an individual's DNA. This is because the number of repeats at a specific loci can vary significantly between individuals. One of the advantages of using STR in forensic analysis is their presence in both coding and non-coding region. Moreover, the analysis is often focused on a specific region of the genome, such as the autosomal DNA, and in the case of YSTR, the Y chromosome, which often pass from the father to son. So, forensic scientists use STR analysis as a powerful tool for DNA profiling and identification. Forensic DNA profiling using STR analysis is widely employed in criminal investigation, paternity testing, and scenarios where accurate identification of individuals based on DNA is crucial.
Now we go to the forensic microbiological analysis. Microbioforensic is a new discipline developed by the Federal Bureau of Investigation. It is based on the fast and accurate detection and identification of microorganisms founded at biological crime with the aim of tracing the source of the microbe. Because microbiological terrorist attack could lead to serious consequences, forensic microbiological analysis has been attracting considerable attention. Further examine the bacteria left by human skin on the surface of contact objects by using next generation sequencing based metagenomic method, and showed that the bacteria left by human skin possess sufficient deoxyribonucleic acid, which known DNA, information for forensic analysis. All the studies described above demonstrate that next generation sequencing has the advantages of high throughput, multiplexing capability, and accuracy, which makes it suitable for rapid whole genome typing of microbial pathogens during forensic or epidemiological investigation. Rare polymorphisms can be reliably detected by analyzing every base of the genome, thus giving forensic data higher resolution and greater accuracy. It is expected that a high-quality forensic microbial database will soon become a reality and aid in the fast and accurate identification of criminals and biological terrorists.
Furthermore, the NGS technology is also applied in animal and plant DNA analysis in the forensic. In wildlife forensics, the application of DNA analysis is widespread for identifying species from confiscated items, a crucial tool in addressing illegal poaching of endangered species cases by examining trace evidence from suspects or crime scenes. This genetic approach is equally effective in detecting species commonly found in the wildlife trade, such as for traditional medicines, hair, and decorative items. Identification process relies on the genetic markers that showcase variability in DNA sequences across species, while maintaining a high level of conservation within a species. Other than that, in forensic anthropology, DNA analysis helps to distinguish between human and non-human DNA and used to establish identity from the mitochondrial DNA in bones and teeth. The current method used for species identification is DNA barcoding, a molecular identification technique that employs a standardized DNA region to distinguish and classify species. The process involves PCR, which is the polymerase chain reaction amplification of the targeted DNA region, followed by sequencing using technologies like next-generation sequencing. A key strength lies in the standardized approach, ensuring global consistency and facilitating collaboration through comprehensive reference databases. However, limitation exists, including potential ineffectiveness for certain taxonomic groups, dependence on the accuracy of reference databases, and more.
So, what are the challenges and limitations in NGS library preparation? One of it is library complexity. This complexity is measured by the number or percentage of duplicate reads that are present in the sequencing data. For your information, duplicate reads means that they are exactly identical or have the exact same step positions when aligned to a reference sequence. The frequency of duplicate reads that occur by chance increases with increasing depth of sequencing. Thus, it is critical to understand under what conditions duplicate read rates represent an accurate measure of this complexity. Not only that, to address the ethical consideration and the privacy concerns of the people of the data, we have an act named the Genetic Information Non-Discrimination Act, which prohibits discrimination by health insurance and employers for genetic information. But, for the biological industry, we have the 21st Century Cures Act, which support initiatives to facilitate the sharing of genomic and other data research and for the clinical use. In the future, we should be using the MySci FGX system, which is the first NGS system designed specifically for forensic genomic application. DNA database organizations are expanding their market size to enable more efficient and collaborative work with global and regional law enforcement. As forensic genomic methods continue to evolve, more dedication can be seen to both supporting and advancing these efforts. There are also ongoing research by the research teams around the world continue to develop new capabilities using NGS technology and forensic casework and human identity testing. Current research efforts are evaluating SIO1003 that provide association with attributes such as shape of the nose, lips, ears, and overall facial morphology, as well as with straight or curly hair.
So, throughout our journey, we have clearly seen the significant and impactful role of next-generation sequencing (NGS) technology in forensics. We emphasize three applications of NGS. First up is the short tandem repeat (STR) analysis, which essentially cracks the code of the unique DNA patterns for identification purposes. Then, there is a forensic microbiological analysis, where we dive into the microbial scene, sniffing out biothreats and exploring microbial ecology. Lastly, we have animal and plant DNA analysis, helping us solve wildlife crimes like illegal trafficking and poaching, also the involvement in forensic anthropology. Thus, with the existence of advanced NGS technology, it can establish a rapid and precise investigation, setting a new standard for forensic science.